In a previous talk, I said that DNA makes RNA, makes proteins is a gross simplification. And you may ask, well, just how much of a gross simplification is it? Well, first of all, let's take the DNA. The DNA is approximately 6 feet of genetic material wrapped into a nucleus within a microscopic cell. So in order for the DNA to be read, it has to be unraveled. I do not have the time to go into all the processes involved in that, nor could I in any reasonable time frame explain it or do the study that would be required to completely understand it. But let's say we have an area of the DNA that's exposed so that RNA can be produced. Now, the DNA does not actually transcribe simple RNA. What it actually transcribes is pre-messenger RNA. So this is the part of the coding region of the DNA that is involved with making a protein. This is what geneticists traditionally called coding DNA. The rest of the DNA has long been considered junk. However, the rest of the process that I'm going to describe to you now is all taking place within the non-coding DNA. And while Dawkins wants to point to junk DNA as evidence of unguided mess, and therefore evidence against a creator, I think you're going to get a different picture when you hear the complexities of what goes on next. So DNA does not actually make RNA. DNA makes pre-messenger RNA. Now, that pre-messenger RNA can be modified by what is called small nuclear RNA. Small nuclear RNA can actually splice the pre-messenger RNA and therefore form different proteins or different variants of the same protein. Once that process has taken place, if it has not been interfered with by microRNA or small interfering RNA, both of which in different ways can silence or slow the production of the messenger RNA, you next require transfer RNA. So the message of the RNA needs transfer RNA in order to make the proteins. The messenger RNA has the information, but the transfer RNA brings into a little machine inside the cell called the ribosome. It brings the actual amino acid building blocks to connect with the messenger RNA, and thereby bringing it into the ribosome can bring the necessary building blocks together. Now, those building blocks are in turn brought together by ribosomal RNA that is combined with ribosomal proteins. They force the amino acids into the right shape. Now, all of those can in turn be influenced by small nuclear RNAs. And again, I pronounce that small nuclear because they are very different from small nuclear RNAs. So small nuclear, messenger RNA, and transfer RNA and ribosomal RNA can all be altered by small nuclear RNAs. And at all of these steps, the protein which is formed or the rate at which the protein is formed can be altered. And so this gives you a little bit of an idea about how complicated this whole process is. And to give you an idea about the relative importance, it takes about between 31 and 41 different classes of transfer RNA to bring the right amino acid or its variant to the ribosome. To give you an idea of the relative importance of these non-coding RNAs, about 80% of the RNA in any given cell is ribosomal RNA. And if any one of these RNAs are eliminated, then the system will not work. And so while Dawkins would like us to believe that non-coding DNA is junk, a position he's stepped back from somewhat in recent years. If he wants to say this is junk, he is sadly mistaken, because each of these different RNA families is required to properly make and regulate protein production. It also brings up another interesting problem for Dawkins and his view of DNA making RNA, making protein, and that is that these other RNA molecules are regulating the DNA. They are actually determining what kind of protein is actually produced. And therefore, the DNA is not the master. The DNA is part of an intricate system of communication that is moving both upward from the DNA, but also downward from the cell, that in turn regulates these mechanisms that determines what ends up coming out as an end product. And so Dawkins' concept of the selfish gene fails to be possible in this scenario, because clearly the DNA is not the sole regulator of the organism that's produced, but it is based on many other factors, as you've been seeing in the last few weeks, many other factors that we are only beginning to understand. If Dawkins wants to use junk DNA as evidence of unguided process creating life, I would challenge that the less junk there is, the more evidence there is for a creator. And again, the more evidence of the proper place of wonder. I want us as people, as a human race, to discover more and more about the way life works. I believe that's what God wants us to do. He has given us minds and He wants us to understand His creation. But to not stand in awe at this incredibly complex process that's going on in each cell of our body at the microscopic level, to not wonder at that, is a grave error. So, once again, please wonder at our amazing God who made it all.